Assalamualaikum warahmatullahi wabarakatuh. Hi, good morning everybody. Let me introduce myself. I am Dwiki Fajar Athaullah, preclinic medical student from Islamic University of Sultan Agung, Semarang, Indonesia. And as you can see on the screen now, I will present you my presentation about our topic today, kinetic engineering. So my talk is particularly relevant to those of you who have a dream to make a world a better place through science and technology. So let's get started. Oh, before we start, I would like to tell you about my table of contents. So in the whole presentation, I divide my presentation into three sections. The first section is philosophy of kinetic engineering. So I will tell you about kinetic engineering using philosophers' point of view. And the second one is CRISPR. It's the revolutionary new technology. And the third, and last but not least, future prospect. In that section, I want you to be optimistic about the future advent after the scientists found the CRISPR technology. So, okay, the section one, philosophy of kinetic engineering. So, okay, let's have a start. I want you all to imagine, I want you all to focus that you are now the philosophers. You have a visions to make a world a better place, but before you change the world, you will begin by asking the question that every philosopher will ask. Can we change our life? It's like the philosophy of life. René Descartes, Aristotle, and etc. So from that question, we can develop it into a several question in our environment. Like, I don't like this kind of environment in this room. So can we change the environment of this room? Or I don't like sour mango. So can I make um, the perfectly sweet mango, etc. So stay focused on imagining that you are the philosophers, and I will recall you to the year of 1980s. And in that year, you will meet some crazy scientists that said in the future, computer will take over everything, that everybody will link with some kind of web, and you own a handheld device that more powerful than nowadays supercomputer. Maybe you will believe, but everybody in that year laughed at him and said that is impossible, that is unachievable. But as you know, all of it happened in the 21st century. So sometimes science fiction became our reality that we don't even think about it. We're at similar point today with kinetic engineering. So let's talk about it. So in this picture, there is the left picture and the right picture, and there's selective breeding in it. So what does it mean? So humans have been engineering life for thousands of years through selective breeding, like this watermelon, from the seedful watermelon, seedful hard watermelon, into the soft, watery, tasty, seedless watermelon. So we pretty good at this. Humans pretty good at this, but I think they don't exactly know what they are doing and how does it work until the scientists found DNA, deoxyribonucleic acid, the code of life, a complex molecule that codes the growth, development, function, and reproduction of everything life. The information is encoded in structure of the molecule, like four nucleotides are paired and make up a code that carries some instructions. So when you change the instructions and you change the being carrying it, you change the person carrying it. And that is kinetic engineering. And this all is the example. And I will tell you some story about some of these pictures, like in the garden, the black and white garden in the left corner. So in the 1960s, scientists bombarded plane with radiations to cause random mutations. Yeah, the idea is for like make them modify themselves with a pure chance. It's sometimes it's work though, but yeah. it's rarely happened. And then in the 1994, the first food modified in the lab, it's called the flavor saffron tomato. It's tomato given much longer shelf life, and we call it GMO nowadays, or genetically modified organism. And in 1990s, babies were made carried genetic from T humans, making them first human with the three parents. And today, we have like super muscle cow or feeder's chicken or eccentric frog, and they make glowing animal too for fun. It's impressive, but it's also with so much money, with so much time, and it's so complicated. And that's all I have to say about genetic engineering. And let's move on to the section two, CRISPR. So, like what I talked about in kinetic engineering before, that is so much expensive, it's pricey, it takes a long time to make, and it's so complicated. But that everything changed when the revolutionary new technology arrived, the CRISPR. Clustered regularly interspaced short palindromic repeats. The cost of engineering have shrunk by 99%, and instead of a year, it's only take a few weeks to conduct, and it's not so complicated. And CRISPR literally has the potential to change the humanity forever. But before we go to the future prospect, let's discuss about how CRISPR works. This is how CRISPR works. There is 6 pictures and 6 captions. So the first one is bacteriophages invasion. So as you know, the bacteriophages, the phages that attack or invade the bacteria, will insert the amino acids into the bacteria and make the bacteria like a factory to make more and more and more of the phages. So commonly, the bacteria will lysis, will shatter apart and become the new viruses. But sometimes the bacteria will survive and activate the most effective antivirus. So the bacteria will save the virus DNA, the part of the virus DNA in the DNA code archive, DNA code archive called CRISPR. So when the virus attack again, the bacteria will transcribe the copy of the RNA of the DNA archive and then put it into the protein called Cas9. So the Cas9 will comparing the DNA with the RNA sample of the virus itself, and it's like scanning all over the bacteria. And when the DNA and the RNA sample is like 100% perfect match, the Cas9 will cut out the virus DNA. So the revolution began when the scientists figured out that this system, this mechanism of the CRISPR is programmable. So you can just give a copy of your DNA you want to modify and put the system into the living cells. And aside from the being precise, cheap, and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and study DNA sequences. It's also work for every type of cell, like human cell, microorganism cell, plants, animals, etc. So the topic is getting more and more interesting, isn't it? And that's all I have to talk about CRISPR Array. But to implement it, what we have studied, I go on the next sections. Last but not least, section 3, future prospect. So HIV, as you know, that HIV nowadays there is no cure to HIV, but in the 2015, scientists used CRISPR to cut the HIV virus out the living cell from the patient in the lab, proving that it was possible. And a year later, there is some tests in the rats that the rats is like 99% full of HIV, and then the scientists injecting CRISPR into the rat style, and it's were able to remove more than 50% of the virus. And I think in a few years, CRISPR therapy might cure HIV. And then there is genetic disease. And then there is cancer. Sorry. So cancer could also been defeated by CRISPR because CRISPR can modify the immune system to clarify cancer better. And the first clinical trial of the CRISPR is early 2016 in US, and then several months later in the August, Chinese announced that they will treat lung cancer with immune system modified CRISPR. And last is modified human. So not only to kill disease, we could also modify our genes to become a modified humans. And for example, if you make your child's brain immune to Alzheimer, why not give them enhanced metabolism or perfect eyesight? And how about the extraordinary intelligence? And even superhuman can be a new standard in the future. But away from that, we need to be careful about this gigantic revolutionary science. It takes a huge amount of responsibility to be implemented. So the summary that I have, I will conclude my presentations into a 4 point. The first one is genetic engineering. It's sometimes called genetic manipulation. It's process of altering the DNA in an organism's genome. Second one is CRISPR, cluster library in the space short palindromic repeats, antivirus system that save part of the DNA in the genetic code in DNA archive. And the third is future prospect. You know, the effectiveness, efficiency of CRISPR can make a better place, can make a world a better place by using it to cure various diseases like HIV/AIDS, cancer, genetic disease, and even modify human. And the fourth one is evolution. We have to be careful about this gigantic revolutionary science, and it takes a huge amount of responsibility to be implemented. So we are going to the last slides. This is my references. If you want to learn more, and I will close my presentation with the quotes from Edward Teller. The science of today is the technology of tomorrow. So if you like this presentation, don't forget to put thumbs up for me and subscribe. And if you have some question, if you have any question, please put the question in the comments below. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.